Hello everyone, this is Dhanashri from Dr. Omic's Research Labs. So, in this session, we are going to discuss about the mapping of reads to the reference genome. As our data is already having a good quality, so we don't need to be perform the trimming. We are going to directly process further for the mapping. And here for the mapping, we are going to be use the tool called HiSat2. HiSet2 you can install by the repository using the command sudo apt-get install HiSet2. But as we are going to map our reads to the reference genome, we didn't downloaded the reference genome yet. As our organism is a Homo sapien, so we need to get the Homo sapien reference genome. But as we are performing this for demo, and if you are going to download a human genome data in your system, if you have a low configurations, it is not going to run the data properly. Also, it is a very time-taking process. So, just to understand the pipeline and for the demo, we are going to download the chromosome 16 as a reference genome. Whenever you are going to perform the real-time data, at that time you can download the whole genome of the Homo sapien. But for now, as a reference, we are going to take only the chromosome 16, only the one chromosome. Why 16? Because whatever the data we have selected, in that data we are going to check the GLC1 knockdown expression. So GLC1 gene is located on 16th chromosome, and that's why we are going to download it. So how to download the chromosome or the reference genome? You can take it from any one of the database like NCBI, UCSC, Ensemble. The choice is completely up to you. So I'm going to download the chromosome from UCSC Genome Browser. So you have to go with the UCSC Genome Browser. Then you can go with the download genome data. Then here the list of organisms is given as we are interested with the human, we are going for the human. Then we are going for the current assembly, that is HG38. And as we just want a single chromosome, so we are going with the second section, sequence data by chromosome. Then if you will just do the control F and you can just search the CHR16, and for the CHR16 you are getting this many hits. So we are not going to download the chromosomes with the alternative and random keywords because there the gene location is not known. So we are going to download the first chromosome, which is your chromosome sequence, like the FASTA extension. So if you want, you can directly download it by just double click. Otherwise, you can download it by using the command. So you just have to right click, copy the link address, and if you want to download the file from the terminal, you can use the command wget-c and you can paste that link. So once you will run this command, the reference chromosome is going to download, and as this is a zip file, so you can use the gunzip command to extract that. You can just give the name, give the command chr6 gunzip chr16.fa.gz, and here you will see that we are having the reference genome which is already downloaded. Once we have downloaded the reference genome, now we can perform the mapping. But mapping contains two steps. So what is the first step of mapping? That is creation of indexed genome. What is the meaning of creation of the index genome or the indexing of genome? Because if you will see that our reads are very short as compared to the reference genome. So whenever we are going to map the reads to the reference genome, it is going to take a lot of time. So what we are going to do is we are just dividing the reference genome into a small files, and they are in a binary format. So as we are dividing the reference genome into a small files and they are in a binary format, so tool can understand the binary language very easily, and it will increase the speed of mapping. The use of indexing is just to increase the speed of mapping. To understand this indexing concept more clearly, let's take an example of book. In book, you have the index, right? So if you want to search a specific chapter, you're not going to search in the page one by one. You're going on the index, you're checking the page number, and you're going to directly jump on that page number. So it will save you a lot of time of searching and it will increase the speed of searching as well. So that's the same logic if you will apply here, that when you are dividing the reference genome and they are in a binary format, so tool can understand the binary language easily, and due to the short sequences or due to the short fragments, it is easy for tool to search the specific location, and this is going to increase the speed of mapping. Now, which tool we are going to use for indexing, that is completely depend upon the tool which we are going to be used for mapping. So here for mapping, we are using the HiSat2, and the HiSat2 tool requires the HiSat2 indexed genome. So here let's prepare the command for the indexing first. We are going to call the tool HiSat2 hyphen build, as we are going to build the reference genome. We are going to build the indexed genome. Then we are going to give our input file, that is chr16.fa, that is our input file, and then you have to give the output file name. So you have to give the same name whatever it is given for the reference genome without the .fasta extension. So this is the command for the indexing, and once you can just copy this command and you can open the terminal from where you are having the reference genome, and once the indexing is done, it is going to generate a total 8 files with .ht2 extension, which means the HiSat2 extension. HiSat2 by default is going to create the 8 files, and these files are the binary files, so you will not able to understand anything from it. So if these 8 files are created, it means your indexing is done. So once the indexing is done, after that, the next step will be we need to map our reads to the indexed genome. So for the mapping, let's open the manual of HiSat2 to understand how to give the command. So if you will just type HiSat2 and enter, it is going to be open the manual, and you can see HiSat2, then option, then in -X you have to give your indexed genome, then you have to give your FASTQ file, and then you have to give the output file, which is either in a BAM format or in a SAM format. So what option we can give for the input? So basically, there is a one option we can give over here, that is DTA cufflinks. So what DTA cufflinks is doing, it is going to report the alignment which is tailored specifically for cufflinks. So actually, as for our next step, we are going to use the cufflinks tool for the abundance count. So the HiSat2 will generate a alignment according to the cufflink tool, so that we can calculate the abundance easily. So that's what we are giving this option. After that, you can give the threading. Means like you can add the threads. Threads means what? It's number of processors. So if you will increase the number of processors, it will increase the speed of alignment. So here, if you will see for -P, there is a option of thread, like a number of alignment threads. So by default, you can run the command without giving a thread on without giving this thread option on one core or one processor. Thread is nothing but what? It's a number of processors. So highest the number of processor, it will increase the speed. Let's prepare the command first. So here I'm just going to call high set 2. Then we are going to add the option DTA cufflinks. Then we are going to give the threads option here, that is hyphen P. Now how many threads we are having for our system, because the configuration is completely depend upon system to system. So how to check? Here you can go with the system information, and in the system information for the processors you can see that I'm having a 8 logical processors. So if I'm giving a 8 out of 8, at that time it is not feasible, because if any another task is running, so at that time we cannot give complete threads. So you have to give the lesser number of threads always. So right now if I'm having 8, I can give 4 or 6. After that, as per the manual, in hyphen X, we have to give our indexed genome, high set to indexed genome. So I'm going to add the option hyphen X, and then you have to give the output file name of indexing, that is CHR16 without the .fasta extension. So once you give the output, once you give the index genome, then you have to give your input file, that is control1.fastq, and then you have to give the output file. So here we are going to generate the output in a BAM format, binary alignment mapper format. So you can give control1.bam. So once you run this command, it is going to generate the alignment or the mapping result. So what kind of result we are getting? Like how many total number of reads are there? So total 12 lakh 50,000 reads are there. Out of 100% are unpaired. Then 95%, approximately 95% aligned 0 times. 3.99% aligned exactly 1 time to the reference, and 0.23% aligned more than 1 time. So here overall mapping rate we are having is a 4.23. So right now, as we are performing this for demo, whatever the mapping rate or the alignment rate you are getting, you can process with that, because that's what the main thing. Once you'll get the alignment, from that alignment we are counting the number of reads, and then we are getting the expression count. So this is how you need to generate the alignment result. Now this command you have to run for all the samples. Like if you have a 4 samples, control 1, control 2, treated 1 and treated 2, you need to run this command for all the samples. So it is going to generate a 4 output files, a 4 BAM files. But the command which I've explained over here, this is what command for a single line data. But in case if you have a paired end data, at that time, how to give the command? So you just have to call the high set 2. Then the same option you can give DTA cufflinks. Then in hyphen P you can give the threads. Hyphen X, that will be your indexed genome. Then you have to give your two input files, two fastq files, forward and reverse. So in hyphen 1 you have to give your forward file, that is sample underscore 1 dot fastq. Then in hyphen 2 you have to give your reverse file, sample underscore 2 dot fastq, and then you have to give your output file. So for paired and data also, it is going to generate only a one alignment file, only a one BAM file. Because though it is a paired and data, it is going to map from forward side or the reverse side. It doesn't matter because it is going to align at the same position. So That's why for the paired data also, it is going to generate only one output file. So from this mapping command, every command will be same for single end and the paired end. So paired data also, like how many number of samples you are having, for that many number of samples you need to run the alignment. And once the BAM file is generated, the next step we are having is the conversion of BAM into sorted BAM. Because the sorting of the BAM file, it ensures that the cufflinks tool, which we are using in the next step, it can effectively or efficiently and accurately analyze the RNA-seq data. But we are going to sort on the basis of what? On the basis of positions of the read, on the basis of chromosome. So right now here we are using just the single chromosome, but based on the position, we need to sort the BAM file, and for that we are going to use the SAM tools. So again, how we are going to install the SAM tools? So you can install it by the repository only, sudo apt-get install SAM tools. So if I will just open the manual of SAM tools, you just have to type the SAM tools and enter. So in the SAM tools, you are having the option of sort. So if you will see SAM tools sort, so it is going to sort the alignment file. So we are just giving the command SAM tools sort. Then you have to give your input file, that is your control1.bam, and then you have to give your output file, that is sorted control1.bam. Now if you will run this command, so it is going to generate a sorted BAM file. If you will see here, this is what the sorted BAM files it is going to generate, and it will sort the reads based on the positions. So as of now, we can't see that what information is present in the BAM file. If you will see here, the sorted control1.bam file is generated, but we can't see like what information is we are getting in the BAM file because it is in a binary format. So that is why if you just want to see like what kind of alignment we are getting, how we are getting the alignment, so at that time you can convert the BAM into SAM just for the visualization. This is completely a optional step. No need to perform that for a large scale data because SAM file required a lot of space. But it just, as of now, we need to understand how we are getting the SAM file, how the reads are aligned. So that's why for one file we are going to discuss that. Otherwise, this step is completely optional. So our next step is what? Conversion of BAM into SAM. And for that, again, we are using the SAM tools. Because in the SAM tools, we are having one option called view. So this view option is used to convert a SAM into BAM and BAM into SAM. So let's prepare the command SAM tools view. Then you have to give your input file, that is your sorted control1.bam, and then you have to give your output file, that is sorted control1.sam. So let's run the command and we will discuss how we are getting the information in the SAM file. So whatever the commands we perform for mapping and for sorting, you have to do it for all the samples, for the control 2, treated 1 and the treated 2 as well. So here the SAM file is generated. Now to see what information we are having in a SAM file, we are going to use the head command because the head command is used to display the first few lines in the file. I want to display the first 50 lines in the file and then sorted control1.sam and enter. So basically SAM file, it is giving you the information in 11 mandatory columns. SAM file here, it's a text-based format that's storing the sequence alignment data from the next generation sequencing experiment. So each line in the SAM file, it is corresponding to the read and that contains 11 columns. So what columns and all it will contain, that we'll see one by one. So this is what our sorted file. Yeah. So here the first column, that is your query name. Query name is nothing but what? It's a name, or you can say it's an identifier of your read which is aligned to the reference. So basically this is what a read ID. The second column you are having, this is about the flag score. So actually this is a bitwise flag which is providing the information about the read, such as like it is whether the read is mapped or if it's a paired end read or if it's mapped to the reverse strand, etc. This kind of information we are getting. So basically this flag is a numerical value which is representing the different properties by adding the specific values together. You don't need to be worried about it. The third column you are getting, that is a chromosome, like to which chromosome or to which reference the read is aligned. That's a reference name. Then fourth column you are getting the position of the base where the read is aligned on the chromosome. So this is what a position. This is a one based position of the alignment on the reference sequence. So if the read is unmapped, maybe if any one of the read is unmapped, at that time you will get the zero value over here. Then next you are getting, that is a mapping quality. Mapping quality, it will tell you how your read is aligned to the reference genome. Then here you are getting the cigar value. So cigar value, it will tell you out of the mapped read how the basis from the read is exactly aligned to the reference genome. So here if you will see, we are getting 143M and 4S, which means like 143 bases got matched and 4 got soft clipped, right? So this is what you are getting the information. Then the next three column, we are having the information about the next read, like this column you're having is the reference name of the next read or the mate read in the paired end alignment. So if the read is not paired or the read is unmapped, so at that time you are getting here the asterisk sign because we are not having the paired end data. Next you are having, that is a position of the next read. So position of the next read in the paired end alignment, and if the next read is unmapped, that is you're getting the zero value. So here you are having the like the next column we are having is a template length. So template length. So it's a observed template length. Means for paired end read, this is the distance between the outermost mapped base of the paired read. A positive value indicating that the mate is downstream and the negative value is indicating the mate is upstream. But if you are not having the mapping information, here also you are getting the 0. Then the next column you are getting the actual sequence of the read. At the last column you will get the Fred quality score, which is represented as a ASCII character. So this is what the ASCII character, which means a Fred quality score. And this is how you are getting the information in the SAM file. This is what the alignment information. And once we perform the alignment, once the alignment of the read to the reference is done, next we need to count like how many number of reads are aligned, and that is what our expression count. So this thing we are going to discuss in the next part. Till then, if you have any question, please let us know. Bye. Have a great day.